Good afternoon, good morning, everyone, wherever you happen to be. I'm Andy Burnett, and I am pleased to be hosting the next in our series of NASA astrobiology road mapping webinars. And today, the lineup of presenters is possibly changing even as we speak, but we've definitely got Aaron Goldman and we've got Andy Ellington, and I'm hoping that we may have Nick Hudd, and I'm going to keep an eye. Say again, Aaron.
Yeah, I'm on.
Nick Hudd signed in now.
Oh, there you go. You see, even as I said, even as we spoke, and there he is.
I was actually on the phone. I was trying to find that start sharing button. Yeah.
What, on the phone? Right.
Yeah.
Okay, so in that case, we have a full complement. We've also got Michael New from NASA's Astrobiology Group, and my colleague Tim Morley over on my right, who is going to be monitoring our chat window. And we're going to do a process of sort of shunting your questions from chat into the live notes. Ah, there we go. Nick, you got your camera working. That's great. And the basic process, just in case you weren't here for the first one that we held last week, which, by the way, is available on YouTube, and you can get to it straight from the front of the website. Basic process is we're going to have an opportunity for the panelists to talk about some of the questions that the NASA team have identified as being particularly interesting for them in terms of the development of this road mapping framework. And then after we've been through that, which might run for 20 minutes, half an hour, we're going to throw it open to everyone who's online with us for Q&A. And then just before we wrap up, I'm going to encourage everyone to go to the astrobiologyfuture.org website, where we've taken a number of the questions that have been identified, more than we'll be covering in this session, and we've dropped them straight in as discussion threads, and I'll make sure to show you that before we end up. So we'd very much like to encourage the conversation, because everything that you discuss prior to the workshop helps to shape the agenda for the workshop. So with that, Michael, do you want to set context for a moment on this particular workshop, on this webinar?
Sure, absolutely. Thank you. So I'm Michael New. I'm the astrobiology discipline scientist at NASA headquarters. Some of you know me personally. Some of you only gotten emails from me, and many of you probably have no idea who I am. We at NASA started communicating with the community about getting this roadmap workshop going a little while ago, and we're trying to cast as broad a net as possible in order to really find the compelling questions that will guide astrobiology for the next decade. The astrobiology roadmap is something we create once a decade, and we update it once every 5 years in order to guide the field as a whole. And when I say that, I mean not just guide what people propose to NASA, but also what NASA is interested in funding as well. So we're trying to cast as wide a net as possible. We want the most creative thoughts and innovative ideas we can get out of this. There's always time later on to winnow that large list down and say, hey, maybe we can't do this in 10 years, or really? That's important? Nah. But in the between, there's a lot of ideas that we want to capture and we want to use. And so this is the start of looking into these questions for the particular area of prebiotic chemistry. We define generally prebiotic chemistry as the chemistry that happens on a planetary surface, so you have to have planets first. And that leads from the basic chemistry on a planetary surface all the way up to the origin of life and some of the early chemical evolution of that life. Now, we can argue a lot about the definition of life, and I don't want to do that on this telecon. And we can argue about where we draw the line at, okay, this is prebiotic chemistry, now we have life and we're going elsewhere. But for now, those are kinds of the parameters. So we're interested in the earliest chemical reactions that produce biomolecules. We're interested in what those biomolecules were or could have been. Looking back in time using both geology and biology, we can maybe see some vague, shadowy outlines of what early life was like, and we'd like to explore that. We'd also like to understand how the environmental and chemical parameters of the early Earth or elsewhere could have shaped what we see now. So that's kind of roughly the playing field, and NASA is mostly stepping back. We want this to be community-led, community-driven. So I'll be here, but you shouldn't hear much from me. Thanks.
Okay, thank you. Let me just build on one point Michael was saying there about really wanting to hear from the whole community. You'll find when we go to the web discussion forum that there are some headings on there. And we got some feedback from last week that people felt a little bit reluctant to start discussions when they thought that they were there on their own, and it was this initial concern about being first person at the party. So we've put in some questions, which the panelists had in fact developed, and we strongly encourage you to chip in your thoughts, because it really is very important that we hear from all the different points of view. And we're also looking at sometime this week, and we'll send out a notice about it, inviting people to come and have just an online chat-based discussion. So using the same Adobe Connect Pro, but if you find you prefer to bounce ideas around when there are other people online, then we'll see if we can schedule that. So we're going to try and use as many different ways as we can. And if you have any technical questions, please go onto the website, just fill in a support ticket, and we will get back to you as soon as possible. So, having said that, let me move on to our panelists and just get some introductions. Now, I'm hoping that Andy is still here, even though his camera has... You are. In that case, Andy, tell us, just for the people who don't know you, who are you, where are you from, why are you interested in this?
Okay. I'm Andy Ellington, University of Texas at Austin. I've been on the periphery of the Origins community for a long time, just like I've been on the periphery of other communities. I maintain a great interest in Origins and a healthy disrespect for most everything that goes on in the community.
And moving, in that case, a natural segue. Nick.
Okay. Hi, I'm Nick Hudd. I'm a nucleic acid biophysical chemist, and my lab is heavily involved in origin research, in particular the origin of nucleic acids and self-assembly.
Great, thank you. Aaron.
I'm Aaron Goldman. I'm a computational biologist, and so I'm interested in looking for those ancient signatures of prebiotic and early evolution.
Brilliant. Okay, what I'm going to do now, hopefully this will advance my slide. Just try that one more time. There we go. I'm not going to read this out, but the NASA team developed just a short rubric saying what they meant by prebiotic evolution. You will find that that's also on the website in the event itself. We've just popped that in there. Well, what we did prior to this meeting was we got the panelists together and asked them to think about what would be the interesting questions that they might want to explore, because the whole purpose of the roadmap is to develop our research questions and the framework to explore over the next decade. So we then sub-selected from that some questions to talk about now, but with the very explicit advice of the panelists that these are in no way meant to constrain their thinking. It's just to give us an opportunity to have a slightly more structured conversation. So I'm going to pop up the questions here. And I'm going to invite the panelists. It's not a case of one by one you give your formal response. It's who wants to start first? This
first one was one that looked verbatim from something I sent, so why don't I go ahead and start off?
Sounds a perfect segue. Go.
Right, so one of the issues I have with the Origins community is that we're looking back into the past at things that are very, very difficult, if not impossible, to really apprehend. And so I think that in order to better understand origins and astrobiology, it behooves us to focus on questions that we can actually answer experimentally whenever possible. And in this regard, I think that NASA is almost a unique community, or the NASA/astrobiology/exobiology/origins community is almost a unique community in really trying to understand fundamental issues regarding replicators. It's not something that NIH does. It's not something that NSF really does. And so I'm very interested in questions about what are different types of replication. I actually consider nucleic acid replicators to be a subset of all possible replicators, and an interesting subset in that they're one of the only ones I know that actually can hold and change information, and those are two very different things. So I'd be interested moving forward in actually developing a framework for how to classify replicators, how to say something about their potential for information carrying and information transfer, and even how such processes of information carrying and information transfer play out in terms of larger functionalities. So there's just sort of a rephrasing of something I already wrote, but that was the motivation for it is I'd like to be as forward-looking as possible and not backward-looking, because I think backward-looking hasn't gotten us as far as we might, at least I might have.
Great. So, Andy, just out of curiosity, what sort of research questions would come to mind?
Right. So, again, any of these tend to be couched in things I know or grants that I would write, and so with that. With that proviso, you know what you know. As you make modified genetic alphabets, what is the fidelity of a modified genetic alphabet? What are the conformational repertoires available to a modified genetic alphabet? To what extent is a modified genetic alphabet capable of mutation? Are there optimal alphabet sizes? What are the parameters that determine optimal alphabet size? How do sloppy replicators compare to faithful replicators in terms of where function arises and how function is transferred, or even how function proceeds across an adaptive landscape? So these are all questions that, for me, I can posit without saying the word origins once, but that I think were very relevant when we had a big mixture of compounds early on Earth, or may run across a big mixture of compounds elsewhere in the universe.
Excellent.
Can I add on to what Andy was saying?
Go right ahead.
So another way that I've heard this put is that that life stores information, genetic information, in a digital way, whereas often the replicators that we create and other forms of information in life are analog. And so I think that within this question, it would also be interesting to study how one kind of replicator can turn into another kind of replicator that might have more potential to be adaptive.
Sure. I mean, I think it. We're going to see that there's a continuum of different types of replicators. For me, it's not an analog-digital continuum. It's more along the lines of fidelity of information transfer and conformational space available to you. I have more of a chemist perspective on this, but I think that absolutely we're going to segue. We can segue one to the other. How those segues occur, when those segues occur, what are the advantages of such a segue, are really interesting questions.
Nick, do you want to chip in?
Yeah. So, Andy, I agree with a lot of what you said. I'm actually wondering, though, whether in this question, and it seems like we have just a few of them, whether it should be broadened or the language should be looked at carefully about replicators. One issue that I have a lot in the origins community has been this focus on a replicator for the replicator, just for the sake of a replicator, and I've had a problem with that because if that thing's going to evolve, I don't see where you can go and get other functions that would be allowed. And so, I've thought that, in addition to thinking about, what has been classically discussed as replicators, again, going to the broadening of the chemistry that could give you that, you could have, say, assemblies that stabilize that are similar to themselves so they last longer or something like that, right? And the process by which they're produced are not necessarily driven by that quote-unquote replicator, but you get the buildup of some molecule, some polymer that becomes a species, and it gets selected because it has some type of an activity, right?
So yeah, this is an argument. This is a discussion you and I had over the years about replicators versus accretors, and I think accretors are cool. I've never quite come up with a theoretical framework for an accretor. Not that it necessarily needs one, but one thing I think we're going to have to do going forward is, how do we bound these problems in ways that the community can understand, or we can just understand, what we're trying to achieve, or even more to the point, when we go out to, say the mission of NASA is X to the public, we can, I think with some reasonable confidence say, by God, we're going to figure out what a replicator is and we'll find one on another planet. An accretor is such a cool idea. It's a neat thing, but I've never really figured out how to do good accretor research. I mean, the best I can see is there's hints of accretor research occurring in the DNA nanotech field, another of the fields that I hang on the edge of, and you're beginning to see some of these issues that you just raised actually answered experimentally, sort of, in that field, but I don't know if those answers are satisfactory for us or for you. I really don't.
I'm not satisfied yet. I'm the perennial optimist that I think that, we will develop experimental systems. And just to throw it out to everybody else that's listening, what are, say, just one example that I could think of that would match this so that we can, put this, a system to the, term accretor. For example, let's say that we have a pool and a chemistry that allows the spontaneous formation of short peptides. And then there's also some type of backward reaction, right, that's hydrolyzing them. And what the accretor might be is that those sequences that can assemble into structures that stabilize themselves from hydrolysis build up, right? And so, if you write out the equation, it's very much akin to a replicator, right, thing. It's templating, but it's grabbing on stuff that's made. And I bring that up because in the language of this, I'd be a little afraid that if it was directed toward replicator, we're going to get everybody trying to get their hypercycle and replication for replication's sake, not necessarily, again, this buildup of functional polymers being selected, right, at the same time that they're being produced.
Yeah, I mean, again, I just think that the thing we want to not do, though, is try and be sitting there juggling 52 grants with 52 different schemas for accretors where we don't have a way to compare them. The nice thing about replicators, when we at least, push on the word information a little bit or push on the word confirmation or function, is we sort of at least have a framework to think about. And even if somebody comes forward with something really weird, we can at least say, okay, well, how would this compare to DNA, or how would this compare to a protein? It doesn't have to be a DNA, it doesn't have to be a protein, but at least we have an intellectual framework for the comparison. For an accretor where we have about 0 examples, I'm just worried people will come in with whack job ideas that, may be interesting but don't really help advance at least what I think of as the field. They don't provide us with something that's really going to give us a better understanding of how an accretor could have functioned at origins or could operate currently in astrobiology in another venue.
Aaron, did you?
Yeah, and this sort of gets back into my transition point that maybe the metric we're looking for is whether it can, if there's a scheme in which an accretor could continuously evolve into something that we would recognize more as a genetic system. That maybe that's the metric by which we can weigh one proposal versus another, is its ability to evolve toward a more complex system like the one that we see. And that actually kind of gets to the last question, so they're starting to overlap a little bit.
Well, let me pick up just at that moment and just talk to the participants who are dialed in on here. We strongly encourage you to add your thoughts into the chat window, just to make sure it's clear for everyone. To the right-hand side of the list of attendees, you'll see a window that's called Open Chat. If you want to, thank you very much, Andy. If you want to add questions, comments, disagree, put in tangential items that you think we should be exploring, do please do that. Great. Thank you, Sandra. Okay, let's move on to our next question. Aaron, you want to start with that? Boundary conditions?
Oh. Well, I'm actually probably one of the worst people to start on that one because I'm
I think that came from my question.
Biologist. I'll let Nick do it. Yeah.
Yeah, that one looks like it evolved from mine. So just to read that is, how is life impacted by elemental boundary conditions? If we had a very different set of metal ions in the environment, how would that impact the functionality of living systems similar to those that have arisen on Earth? So in the background of this, before we had sent in a number of questions, and I think that this one is probably a summation, or came out of a couple of different ones I had in there. And I think that it's nice that the first part of that has to do with the metal ions, because one big point I think that we have to contend with in original life scenarios is that the early Earth clearly had a lot more iron 2 available, right? It's all been precipitated in this banded iron formation. And if you were to point to one metal that would affect chemistry, right, iron's a good one. I mean, it's an amazing participant in redox reaction. We use it all the time in life today. So it begs the question of, if we're doing prebiotic chemistry research, how different would that be in an oxygen-free, iron 2 rich environment compared to what we're doing in the lab? So I think that we have to ask those questions. I don't think they've been asked enough. And expanding that too, beyond just the iron question, we can think about, again, different scenarios having to do with the atmosphere. Was it really high CO2? How does that affect the atmosphere? Was it reducing? Do we have H2 available? I think we got to think about all those parameters and not just give carte blanche that you can do anything you want, but it's got to be connected to a scenario that shows that this provides interesting chemistry or interesting ways out of a present quandary if we consider alternative conditions.
If you were going to spend research money on this question, what sort of research programs would you be interested in seeing? So this is an interesting question, and I'll start it with just the same way that Andy said. When you look at this, you look at it for your own research, right? And so you think, this is what I'm doing, and I like this, and so preface it that way. One thing that we're doing right now is we're looking at in collaboration with my colleague Lauren Williams, we're looking at what can RNA do when it has iron bound to it rather than magnesium. And the answer is it can actually do a lot of chemistry it can't do before that. And so that means that we could potentially solve a lot of problems that now are solved by proteins, with RNA binding iron. So looking at these kind of what if questions of what if the system we had today was in that alternate environment, what could it do or could it not do, right? And so that, to me, and I think that connects somewhat nicely with what Andy had mentioned. Andy says, what is the experimental system that we can use? We don't know everything about what the environment was like or what molecules were like, but we can take things we have to extrapolate back. And I think that those are really good questions to ask. With the biopolymers we have today, under the alternative conditions, maybe the minerals, the solvents, the ions, the atmosphere, all different, and so that could give us some insights on how they might have been able to function differently, constraints on whether they really did start at those times, constraints on what the environment must have been at the time that they did emerge.
Great. Possibly, broader than this question would be which aspects of biology and biochemistry are not only elemental boundary are impacted by the elemental boundary conditions, but also just the historical contingencies of life's origin. So, for example, the main energy currency of life is also the main subunit of the genetic molecule, and is that because that molecule is really good at both, or is that because it can do both, and the origin of life just so happened to favor one molecule doing both things?
Andy, do you want to add anything on this?
Yeah, so I think a slightly different view than Nick or perhaps some of the other participants. I'm not particularly concerned about the boundary conditions for Earth. I'm really, again, much more interested in novel chemistry. How do different chemistries, promote the origin and evolution of any of a variety of living systems? And for living systems, I generally confine myself to replicators rather than accretors, but we can make it broader as necessary. So the problem is, of course, and I think Nick did point this out, that we don't have a whole heck of a lot of examples. But what I could see being funded out of this would be anything from, say, Lee Cronin's molybdenum replicators that he's been mucking about with over in Glasgow, up through folks who wanted to expand the genetic code to 256 amino acids. And while the latter is definitively post-origins, nonetheless, it is a system that allows us to explore what chemistries help or don't help. If we had more selenium in the environment, what redox chemistries would cells evolve? Could they evolve? If we remove potassium from the environment so that we can go on very long space journeys without K40 eating us up, what would those cells be like? So I think that cells give us a nice system for exploring changes in chemistry that affects living systems. And other than that, we're going to have to either, again, change genetic alphabets or other sort of replicator-based chemistries, or come up with really cool new chemistries, again, a la molybdenum or other replicators that almost defy description at this point.
So let me come in there and kind of perhaps disagree a bit with Andy there, is that within the charge of the program on prebiotic evolution, I'm not so sure that you want to just open it up to what replicator of anything, right? Because I think that the ultimate goal of this is to see how life on Earth could get going or got going, and can we have similar life on other places? I mean, if you broaden it up, like you say that is Lee Cronin's system living or not. The problem is that the space is so broad in chemistry, or chemistry space rather is so broad, that I think then we get into this, well, is this living or is this not? I think we're better off if we are looking at systems that are connected to what we had today, or we think are potential ancestors that we had, and then asking the question, how could the environment have facilitated this on the early Earth, or how could your favorite scenario, would have it been allowed or disallowed by what we know? Now, the constraints are, still being formulated for the early Earth, and George Cody had put into the chat box there, something about geochemical constraints and elemental availability, and as he said, like phosphorus is still a problem, right? Or is a problem in the absence of life. So these I think are big questions that are already broad enough without saying we'll let life be whatever you want to define it.
Yeah. So again, it unsurprisingly, Nick, and again, this is part of a long, ongoing conversation. I do disagree with that. It's up to NASA, of course, whether this is going to be solely related to Earth origins or not. But I think that stricture in one hand, it's a little odd coming from somebody who was just arguing for accretors, of which we have no modern examples. But nonetheless, if we're going to restrict ourselves to sort of terrestrial chemistry and biochemistry, I think we do a disservice. The field has focused on that for a long, long time, and I would argue without a whole hell of a lot of success. And one of the reasons for that is not that they're not really smart people working really hard, but because I think many of these questions are ultimately unanswerable with respect to terrestrial systems. We just will never know. And I think by broadening it to the possibilities for living systems here or elsewhere of any of a variety of stripes, we give NASA a broader, better, more answerable mission rather than just retreading, the origin / exobiology / astrobiology rebranding that has occurred so far. So if anything, I would like to go exactly the opposite direction. I would like to just ignore terrestrial constraints to the greatest extent possible. But again, that's just my own opinion. NASA and others will do what they want.
Well, I disagree that we haven't made any that we haven't accomplished anything in the last 20 or 30 years of doing this research. And I think that looking at modern biology, both looking back through modern biology and understanding what order, at least we think, to a very high degree of confidence, the different polymers arose, and then also looking across biology to see at least what the constraints are in modern life has been valuable, and we can't really toss that away because it's our only example wherein we know that life did originate. I agree with you that we should, that this should be a discussion, in my opinion, about origins of life throughout the universe. But we have this nice example, and it's been productive looking at it.
Okay, let's move on, and in the meantime, I'll email Lee and ask him to send us all his 3D reaction-ware diagrams so we can print it out and do our experiments while we're talking. All right, minerals. Now, Nick, should I start with you again? Role of, if any, of minerals could have played in early catalysis?
Yeah, I think that one actually was probably right out of my questions on there. It's a reoccurring question, right? We know that life probably evolved in contact with some type of substrate, and just as Michael had said, they're not interested in life starting out in maybe the gas phase. It's on a planet, right? And if you're on a planet, you're on a surface, and if you're there getting life started for millions of years, there's probably some type of interaction going on with what was the substrate. And I'll be the first one to say that I'm not really into this really strong connection to the minerals where you have like the clay replicator or something like that. But that being said, minerals are absolutely a source, a sink of ions that could have been important. They're where you're going to find your phosphate, if phosphate's where that element is going, the phosphorus is going to come in from. So you have to think about that. And then, of course, minerals can act as catalysts, and there's been a lot of theories on that, like the iron sulfur ones. And so I just think it's still something that's worth considering for sure.
So let me ask you, based obviously on my very limited understanding of this, has any of the conversation that we've explored so far involved sending probes to Titan? And the reason I ask that is in the previous webinar, which was about exploring solar systems, people were very keen on all sorts of missions. In terms of identifying roadmap sort of questions and where NASA should be spending its money, what does prebiotic chemistry actually require? Is it all lab-based work?
One presumes there's some component of astronomical observation there, that understanding the chemistry that occurs elsewhere as best we can informs what could occur on a planet or wherever, or what we might want to attempt in a lab. So I would say that it doesn't have to only be experimental. It could be at some level observational.
So what I'm really interested here is Nick, if we take what you've been talking about, say, in terms of the minerals, are there particular research questions that if you're thinking in a decadal terms, you'd say, this is what I really want to know? And obviously this is a question to all three of you. Where would you spend your money, given that you have a bias to your own areas of knowledge?
Yeah, I think with respect to for just as an example on the phosphorus one, right? That's just if you don't have it, you don't go to that next stage. So where was the phosphorus? How do you get that phosphorus out, right? And use it in the earliest systems. That's an example. And then what came up in the chat window is that the study of ancient terrestrial rocks could add some context. I think that's an important thing too, because if you're what we know quite well is that there's an incredible evolution of the rocks that we have on Earth, that there was far fewer, as Bob Hazen pointed out, species early on. And so you want to say, okay, given what was there and what we see as the critical reactions to make certain molecules, could some of those play the roles? I think very specific questions like that can be asked.
I would add, from the top-down perspective, there's a number of very ancient proteins that are central to areas of metabolism that we know are very old. DNA manufacture or the generation of energy through the membrane-bound ATPase. The proteins associated with these functions, their catalytic core is made up of inorganic cofactors that in many ways mimic the structure of iron sulfur minerals, and organic cofactors that are derived from nucleotides. Now, that could easily be a coincidence, but it's a very interesting lead, and I would like to see that pursued in more detail.
Just picking up a question from the chat window, Michael. So the question being asked here is whether the window of the roadmap is to guide research indefinitely and updated every 5 years, or whether we're mapping research ultimately, what comes out of the workshop. Is it mapping research for the next 5 years?
Well, so what I wrote in the chat window is, while it's true the time window of this roadmap is expected to be about a decade, not 5 years, a decade. And yeah, at 5 years out, we will reassess and tweak it a little bit, but we're not expecting massive structural changes. So what we say in this roadmap will exist for 10 years. That being said, Mary and I and NASA would very much like this roadmap to be very inspirational and aspirational. And so it's much, much better in my mind, and I think in NASA's mind, to have a document that thinks much bigger than 10 years than a document that thinks much smaller than 10 years. Once you have a bigger arc laid out, it's easier to go in and talk tactical changes. Should we do this experiment or that experiment? It's much harder if you have just a list of experiments to do to then say, all right, well, so we've done these experiments, now what does that mean for us? So we're definitely looking for maybe not an infinitely sustaining program, but a document that will guide us for at least 10 years, probably more.
Brilliant. Okay, thank you. Let's go to our last question, though, and then I'll open it up for the audience to ask questions. Aaron, life evolving through a series of biopolymers. What were your thoughts?
Right. This is my favorite of the four, actually. So thanks for choosing me to go first. So I think this is we're starting to piece together at least the later, what I would consider to be the later stages. That originally there might have been more polymers. Like, for example, it seems like it's easier to get peptides than small RNAs chemically, but it seems like RNA was the original, or not the original, sorry. At one stage, RNA was both the genetic and functional molecule and might have used other functions as well, but it was the main chromosomal molecule. Then began to encode proteins. Then proteins allowed the life form to build a large DNA chromosome. I think this is one of, it's not necessarily too recent, but it's one of the major successes in the field of the last couple decades. And so the sort of work that Nick does, looking at alternate polymers that could have preceded this system, I think is very important. I gather the work that Andy Ellington does trying to expand this system, I could see as very important. And I'm interested in what the selection pressures are to start increasing something more, start to remove the genomic molecule from the to separate the genomic molecule and the functional molecule, and then to, as in RNAs and proteins, what are the selection pressures that drive an organism to do that, and how very early on did something as elaborate as the translation system arise that allowed for that to take place? So that's what I'm mostly interested in.
Thank you. Nick, what would you add to that?
This is a question I like a lot, very near and dear to my heart for sure. I'd add that when I look at this, I also like to think about what is the simplest polymers. This is just really kind of reiterated some of what Aaron said, but from material availability, from chemical reaction, what are the simplest polymers that could have come about that could have evolved into the ones that we have today? I think that that's a really, really important question. I think that most of us would agree that the spontaneous formation of a long functional or even replicating RNA is not likely. So there were stages that built up to that. And so I think that connecting that to experimental systems and showing assembly of rudimentary proto-RNA is important. On the peptide side, it's still not even a solved problem of how do you make oligopeptides, 5-mers, 6-mers in good yield in a prebiotic system. And so maybe esters came before that. Can we come up with robust chemistries that give us kind of a foothold onto informational polymers that then can evolve into the ones we have today? And then, this cooperation between polymers. The word cooperation didn't make it in there, but Aaron talked about kind of a series of one polymer giving rise to the next. And I think that what's important also to think about is that we might not have had this kind of serial development of the polymers. In fact, I think there was a whole lot of them working along in parallel and evolving, and then major steps in evolution may have been when certain polymers started to cooperate, like peptides with nucleic acids with membranes being another polymer of aliphatic molecules. So that's what I just add to that. I think it's captured in there, but maybe it should be expanded a little bit more about cooperation between the different types of polymers.
Brilliant. Andy, any
thoughts? So I think that we're sort of saying much the same thing, except whether or not we have a historical focus. So what kind of alphabets or chemistries we have are important. What the potentialities of those alphabets or chemistries are is important. The transitions between them is important. We either have a timeline that goes into the past and is constrained by terrestrial boundary conditions, or we have multiple timelines that are occurring elsewhere or that go into the future. And I'm more in favor of the broader view, but it seems like we do sort of speak about the same questions. The transitions are interesting because I think even as George pointed out in chat there that the ribosome sort of constrains our thinking in a way. The ribosome freaks me out. I mean, I just don't even like to think about it. Translation is just weird. And yet it occurred, and it occurred in some context. And maybe, 20 billion years from now, somebody, the Nick Hood of Alpha Centauri will be arguing about why we have these, silicon organic things where the organic parts are this weird heterocyclic compound, and where did that come from? I mean, I could imagine, further transitions in our own evolution where it would be hard to look backwards and see how it happened. So I just sort of disregard translation because it's just too weird, and I don't think I'm ever going to know enough to figure out anything about where it came from. So, these transitions can be as stark as translation or as silicon organic, chimeras, or they can be, milder transitions, PNA to DNA, or ester to phosphate, or whatever. And these are all worth studying. I think that we should just study them in a context where we have a common language and a common way to compare what it is we're trying to accomplish. I think that's more than anything what I'd like to come from some of these discussions is that we can speak together as a community and have ways to compare our different ideas no matter what we're working on.
Okay, well, at that, let me pick up on the idea of speaking as a community. We have a few minutes left on this session, and I'm conscious that I don't want to overrun because people have given us their time. If anyone wants to ask a question, Mike is going to open all the mics. At the top of your screen, over here on mine, you'll find a little icon of a person with their hand held up, and it seemed to work quite well last time. If you want to ask a question, make a point, just click on that, and your name will pop to the top of the list, and then we will go from there. We encourage people to vehemently disagree as well. So taking the broad topic of prebiotics, what are your thoughts? In that case, if the panelists have covered the subject in such perfect detail, Mike, can I ask you to bring up the web browser window, please? Great. It's possible that that some of you may not be registered on the website or may not even know about it if you discovered about this webinar through another route. So I just wanted to show you very briefly, thank you, the website address, astrobiologyfuture.org. And then, Mike, if you scroll down ever so slightly, you'll see that in the middle of the screen now, it says share your thoughts. And just below that, it says click on this link. So I'm just going to show you. If we click there for a moment, what we have now is we've set up 3 discussion areas. The advanced life one is for our 3 o'clock East Coast session coming up soon. Do not miss it. But further down, we've got our prebiotic life one. And at the moment, we've just got 2 sections there, ideas and questions. You'll see that for the solar system exploration one above, we also have the transcript from the meeting chat, and this will be added in here as well. But what I'd like to do is to encourage people to go over to the site, go to the prebiotic life section, and particularly on the questions there is a larger collection of questions, and it's actually going to be updated shortly. I just got a couple more from the panelists. And please go and add your thoughts in that. Now I can see someone has already. So that's what we're going to do. I did see one question appearing in the chat. Oh, yes, Sanjoy, about actually several while I was talking. All right, let me make a suggestion in that case. If there is a question here that you want to have covered, and our panelists are going to be engaged with the site for the next week or so, feel free to add your own question into the prebiotic. And when you're doing that, please take the time to add your thoughts to this. It really is very important to help NASA shape their workshop and the resulting roadmap. And with that, I think we will draw it to an end unless anyone wants to stick a hand in the air in their last moment. And
Mike, what is advanced life? Have we inadvertently tread upon your other session? I don't quite understand the distinction.
Oh, sorry. Yeah, we tend to use evolution of advanced life to talk about a couple of things. The title comes from looking at the transition from unicellular life to multicellular life and all of the evolutionary changes that occurred in that process. We also in there tend to include the response of the causes and the responses of ecosystems to mass extinction events, impact, flood volcanism, that kind of thing. So it sort of squishes together issues in developmental evolutionary biology and issues in sort of macroevolutionary paleontology.
Okay, so our various discussions of, the role of chemistry in either origins or the expansion of functionality are perfectly legitimate under the prebiotic banner.
Absolutely, and they're probably more easily tackled. I don't know. It's a question whether they're more easily tackled in the prebiotic milieu or in the advanced life milieu where you've already got a fairly complex system of genetic control already established.
I'd be concerned that some of the fundamental issues would fall through the cracks, so I would like to err on the side of complex prebiotic evolution.
I didn't mean to imply that prebiotic evolution was simple. Just that we tend to think of unicellular life and multicellular life, and historically multicellular life animals have been considered advanced life, and so that's where we grab the words. If you think of a better title, I'd be happy to accept them, honestly.
Sorry, I was actually referring to next week's discussion on early evolution, which probably will have a lot of crossover.
So early evolution generally means once you've got a living system, what happens?
Okay.
That's kind of arbitrarily where we draw the border. But my comment stands. If you have better titles for these things, I would happily welcome them.
Okay, just 2 points. Damien had raised his hand just while I was talking. Now, he's added the question into the chat window, and I can see that Andy's responded, but just for Aaron and Nick, so he's asking, the panel talked a bit about replicators in general. Any general model is defined by a set of constraints, and what the model does not model. What would the panelists see as the criteria for such a model?
Yeah, I think so. Sorry, go ahead.
So I think that's actually a really good point, because this is goes back to I think that Andy and my definition of these systems that he was calling accretors are a bit different, and he says there isn't an example of those out there. And I'll just throw out, an example I see in a sense is the propagation of peptides that form amyloids is all over the scientific literature. And, some of the models for why do these build up is that the proteases cannot cleave up peptides in the amyloids as well as they can free peptides. And so when Andy talks about accretor, it sounds like he's talking about one single molecule entity, and I'm talking about them coming together and doing something that promotes their own survival, and that's a type of self-selection. And so, that's where, with respect to replicator, pretty much everybody has, in the modeling said, this molecule gives rise to a copy of itself. Well, okay, that's strictly speaking, that's a replication, but we could also have a broader sense of it where oligos are being produced, and then by their association, they are then living on, a longer time, and that's a selectionist too. And so that's a little broader definition, perhaps, of replication. Okay.
And
I think that's a great example.
Sorry.
Sorry, go ahead.
I was just going to say I agree with both Nick and Andy, and would only add that for me it is more important that it's capable of evolving in a manner that we would recognize as biological evolution.
Okay, I am going to draw it to a close now, but I just wanted to address Aaron's question about how does the information from the discussion and the chat windows feed into the roadmap workshop. And the answer is that we're using these webinars and the website to broaden the discussion in the sort of more diverse community, to give insights to the questions that might be part of the road mapping workshop. So really at this moment, all we're doing is data gathering in order to allow us to get together for 3-ish days to have the academic debate. So that's the purpose of both the webinars and the website. Okay. So with that, thank you very much, everyone. I will see some of you, I hope, possibly all of you, in an hour or so at our 3 o'clock session on Advanced Life. But thanks very much. Cheerio.